So hello everyone. I would like first to thank the organizer for giving me the opportunity to talk about this great subject, that is the genomic contamination detection, and to such a large audience. So I will begin my talk by showing what is a genomic contamination. So here on the top, you have one genome, highly contaminated, and on the bottom, another one who is only slightly contaminated. And it permits me to introduce you to specific naming that I will use for the rest of the presentation, and that also use in different publication. In green here, for the slightly contaminated one, the contig that belong to the organism that is linked to the genome is the contig from the expected organism. So when you download the genome somewhere from NCBI or another repository, you have a taxonomy associated to him, and this taxonomy is the expected organism. From time to time, it's not the main organism, and it happens that the expected organism is simply lacking from the genome and is replaced by something else. Here you have in red what is not linked to your expected organism and who is the contaminant. And you can see here that it's a very clear case with a different GC content, a different coverage, and here you have a different taxonomy also for the ribosomal gene proteins and the 16S. So that's how you can represent a genomic contamination. How is it possible to have in your genome something that you don't want? So we made a literature about different cases, and what we notice is that it can happen nearly everywhere since the collection of your organism or your culture until the use in silico or computational use. But anyway, if independently of the different origins, it can be summarized into 3 cases at the sequence level. So you have 2 big differences, the one that are redundant and the one that are non-redundant. So the redundant contamination is when you have a gene that is from the contaminant that is already present in the sequence from your expected organism. That's the redundancy. When this gene is not already present, that's the non-redundant contamination. And you have 2 differences here. If the genes was naturally present in the expected organism, but is replaced by the contaminant one, that's a replacement. And if not, it's a single contamination if the gene was not naturally present in the organism. And so this contamination can throw into database, and you can, of course, found it in the genome that you download. How often it happens? Here you have an overview of RefSec that we made 1 year ago, and you have the different taxa. Here the bar plot indicates the number of contamination that you can find in the genome. In light is when it's contaminated below 5%, and in dark, like here, or like here, is when it's contaminated by more than 5%, so highly contaminated. And 2 interesting things to see from this plot. So we can find contamination for all the taxa present in RefSec, sometimes with a high number. But what is also interesting is that you find here different. For example, in green is the leader of the market, CheckM, and in blue is our own tool. And what you can find is that sometimes we found contamination and CheckM not, and sometimes it's the opposite. So how is it possible for the same phenomenon to have different measurements? That's what I would like to highlight with this slide. So we made a classification of all the tools that are available for genomic contamination detection. The tool on the left here are the one who need more user interaction and who are more cases per cases. The most used tool by the community are the reference database linked to the one linked to the reference database, sorry. And here you have 2 big category. The one who estimate contamination based on duplication of gene markers, like CheckM or Busco, or the one that are based on the whole genome, genome-wide, where you can find CRACOT, who is an exact gamma matcher, and also those who are linked to BLAST, like Gunsey, a very recent tool who measure taxonomic chimerism on contigs, and my own tool, Physeter, who measure contamination based on BLAST report, on last common ancestor on BLAST report. So you see that you have different taxonomies. So that means that when you estimate contamination, all these tools estimate contamination, but you don't measure the same thing. This is why you have such difference when you try to do with multiple tools. And we did that for cyanobacteria a few years ago with a lot of tools, and it was really impossible to correlate them and to correlate the results because of this difference. So what we would like to know if it is what is the precision actually of this tool? Can I rely to him? And which tool should I use for my analysis? This is what we try to answer with a tool called CRACOT, where we simulate the 3 types of contamination that I told you at the beginning of the presentation. So for that, we use the orthology inference with a selection of very high quality genome that we are sure that are not contaminated to simulate contamination and to be sure about the amount of contaminant that are integrated into a simulation. And for that, we use orthologous groups. When we have a group that is shared by multiple genome, we take only the groups that are unicopy. And so it made the common OG that I use to create replacement contamination and duplication contamination. And then the singleton I use to create single contamination. So that's the 3 cases that we simulate: redundant, replaced, and single. And we run multiple tools on this simulation, and we made the simulation at different taxonomic rank to have an idea about the efficiency with an increasing difficulty on the taxonomy. On redundant contamination, what we discover here in this plot, where in blue line correspond to the level of contaminant from the simulation. So the blue line is our simulation. And what we can see is that frequently for CheckM, Busco, also CheckM2, and also for my tool Physeter, it's over-detected, largely over-detected. And the only tool that actually fit well to the simulation is CRACOT2. You can see that in red you have the Spearman correlation, and you can see that it is really badly correlated and for sometimes negatively correlated to the contamination level of our simulation. When we switch to replace contamination, that's the opposite. Logically, the tools who are based on duplication of markers like CheckM and Busco will over-detect the contamination. But it's also the case for Gunsey, not the case for Physeter, who is still in over-detection. And again, the only tool that fit well to the simulation is CRACOT, at the exception of the species level. We can also note here that at the genus and species level, Gunsey cannot detect contamination. The result is the same for single contamination, who is largely over-detected by the tools, at the exception of Physeter, who is still in over-detection. And again, CRACOT is the only tool to fit well to the simulation. So this is my last slide. My take-home message is that, so what I show you here is a combination of 4 articles that we published since 4 years on this subject. And we know that there are different types of contamination and a multiple tool that allow you to detect them. And each tool has its own advantage, and users should be aware that they need to not select one tool, but multiple tools that are complementary to be able to catch contamination. In the actual state of what our simulation show, it indicates novel detection of redundant contamination, but a non-detection of replace and single contamination, who might also occur in the same frequency as the other one. And also genus and species rank are the ones that are the most complicated to detect. So I will finish my talk by thank my advisor and my collaborators on this project. Thank you.